Hi, my name is Melanie, and I'm a member of the Zeitlinger Lab at the Stowers Institute for Medical Research. And I'm here today to talk to you about our work as biologists and computational scientists leveraging deep learning, much like the Oracle of Delphi would impart predictions upon her supplicants that were very accurate, but also very confusing and often shrouded in mystery. We leverage neural networks in many similar ways because we train it on data that we feel like we understand, and it imparts very accurate predictions. However, the reasons behind those in the cis-regulatory syntax rules is often hard to decipher, and it's only through the merging of biology and computational science that we can begin to discover really subtle things and cool things about how our genome is encoded. Enhancers are regions of the genome which are known to regulate gene expression. Add enhancers are short DNA sequences called motifs which recruit transcription factor binding. Through the combinatorial arrangement of enhancers, this is what allows the enhancers to be driven to a more active state. But in order to understand the sequence logic, we first need to ensure that all the motifs have been correctly identified. Traditionally, we've done this using position weight matrices. And in the event of a close sequence match across the genome to a consensus PWM matrix, we can consider that motif to be high affinity. Clean match, clean matrix, it's all good. However, in the case of degenerate sequences, it is often very hard to distinguish false positives from actual functional low affinity motifs. And recent work has really shown that low affinity motifs can be really contextually crucial in development and expression of certain genes. So my work surrounds the question, if low affinity motifs matter, in what fashion do they matter? And I've chosen to bound this work specifically to one step of cis regulation, in which at enhancers, chromatin usually starts out as closed and inactive. Through the binding of pioneering transcription factors to their consensus motifs, they're able to remodel the nucleosome wraps DNA in such a way that the chromatin can become accessible and the enhancer now can be driven to a more active state. My question is twofold. If we can find the low affinity motifs in the first place, is do they mediate pioneering events? And if so, do they participate in combinatorial enhancer logic? Low affinity motifs, by definition, only weakly recruit transcription factor binding. So are they functional enough to actually play a part in the cooperative sequence syntax? Now, deep learning is uniquely suited to this because we've previously shown that it can learn contextual sequence rules of transcription factor binding. And we suspect that it is this surrounding genomic context which lends function to these weak motif sequences. Here, I show a previously published convolutional neural network called BPNet. The inputs are DNA sequence, and the outputs can be actually quite a variety of genomics data. Here, I trained a neural network on about 150,000 sequences from bound and accessible regions in the mouse genome to explain the chip nexus binding profiles of 5 different transcription factors, 2 of which are well understood pioneers in mouse embryonic stem cells. We chose this model system because we understand the sequence syntax rules quite well, and this is a well-studied system. So if our model's rules align with known existing literature, this would be an excellent validation to kind of jumpstart our interpretations on what the model has learned. Additionally, if I really want to nail whether low affinity motifs act as pioneers, it would be useful to have a sequence to accessibility relationship captured by another convolutional neural network. Here I show a published representation of Chrome BPNet, which rather than answering the question of where transcription factors bind, it uses a modified training paradigm to answer the question of how open is the DNA, with output data being ATAC-seq data with the TN5 enzymatic bias impacting where the position of the cuts are removed from the output predictions. Thus, if the sequence rules are shared across these models, this would serve as independent validation that the sequence rules that we're capturing are indeed indicative of pioneering events. So I'm going to cover this pretty briefly, but we trained a bunch of different models. Our model performance was comparable to the same data published in 2021. And under different training paradigms, different data sets, the models were able to be reproducibly return the same kind of sequence logic, which for us is the absolute most important thing downstream when we're interpreting these models and their relative performance. I show here a showcase example of a genomic region that is important for pluripotency maintenance in mouse embryonic stem cells. And we can see that the observed and predicted binding and accessibility profiles match quite nicely, which is a great human brain litmus test of whether these models performed well, which is why I'm showing it here. After ensuring that our models performed well, we then leveraged a contribution or attribution assignment technique called Deep SHAP, which explains each nucleotide's contribution to the output predicted signal. So thus, the higher the contribution score, the more it's explaining the output predicted either binding profile at the top or the accessibility profile at the bottom. After we found all the regions that are super contributing to either binding or accessibility, we leveraged a tool called TF-MODISCO to consolidate high contribution into motif representations for both our binding model and our accessibility model. You can see on the right our 5 main motifs of interest: OCT-SOX, SOX2, KLF4, ZIC3, and NANOG are kind of the key players that we were interested in this MESC model system. And it actually seems like, for the most part, both the binding and accessibility model both returned contributing motif representations. However, interestingly, the NANOG motif was not found to be important for the accessibility model. And a paper published last year actually reported in MESC specifically that NANOG was not found to be a pioneer. So the alignment of recent literature with our models show that we really indeed are learning the right sequence rules in vivo, which was encouraging us to dig a little bit deeper. So like I mentioned before, I want to look at low affinity motifs. So in order to do that, I need to map them back to the genome somehow in a high confidence fashion. Like I mentioned before, traditional approaches just use local sequence match, which, while pretty powerful, is really hard to distinguish non-functional false positives from functional low affinity motifs. Our previous approach in 2021 leveraged both a sequence match score along with contribution metrics to filter out these false positives and rely on model contribution assignments to figure out whether a motif was deemed to be important. Here, in my current approach, because I wanted as many degenerate low affinity motifs as possible to see what they were doing in vivo, I removed the sequence match criteria because I really didn't care how degenerate they got as long as the motif generally thought that they were functional and for the right transcription factor. So when I did that, I got about 25% more motifs. So the next thing I needed to do was validate that they actually were functionally bound degenerate motif sequences. So here I show OCT-SOX motifs important for the binding model specifically. I show both the top high affinity motifs and the worst low affinity motifs. And we actually, in order to validate whether they were bound, we leveraged chip nexus experiments. Chip nexus is unique in that it provides sharp base resolution maps of where the transcription factor is bound. So indicated by the black arrows, you can see that for at least the OCT4 transcription factor binding protein across its consensus motif, at the low affinity motifs, even though it's weaker bound, which is what we would expect, we still see these distinct footprinting patterns indicating that it is weakly bound, but it is specifically bound. Additionally, though the accessibility model had never before seen transcription factor binding data, it also was able to return the OCT-SOX motif and at the best and worst sequences produced a sharp footprinting profile, raising our confidence that these low affinity motif locations could actually be interpreted as functional in vivo. And as a benchmarking, I also performed PWM scanning across the exact same regions that I mapped using model contribution methods, returned over 500,000 hits, and of those, only about 40,000 or 38,000 of those were deemed to be important by our models. When I show the OCT4 binding in gray across the best matched PWM unique sequences, we can see there's almost no binding, indicating that the models are indeed at the level of DNA sequence able to learn functionally bound sites that are actually quite degenerate. So after confirming that I could find low affinity motifs, I next wanted to ask whether the models thought they were predicted to mediate pioneering. People call these a lot of different things. I'm calling them isolation scores for the sake of colloquialism, but Peter Koo has coined them as global importance scores. We've previously called them marginalization scores, but they're essentially proxies, like in an in silico environment, of whether it will have an effect on transcription factor binding. Here, we take all the unique motif sequences, we inject them into random in silico sequences and measure the log fold change effect either on the binding model on the x-axis of these scatter plots or on the accessibility model on the y-axis of these scatter plots. Here, we immediately see for both the OCT-SOX and SOX2 motifs that there's a nice three-way correlation between binding, accessibility, and motif strength, which matches our previously published expectations of what would be a pioneer. So at least in silico, they seem to be acting as pioneers, albeit more weaker pioneers, which would be our expectation. So the next question would be, do they participate in combinatorial enhancer logic? Can they cooperate with other motifs in the enhancer in order to increase accessibility? So here what I did was I took every single motif pair of all of our motifs of interest, I combinatorially mutated them and predicted the effects in our accessibility model. We decided to characterize cooperativity fairly simply, and simply asking, is the effect of the motifs together greater than the individual sum of their parts? Just an additive model. And we can immediately see that in the red parts of the heat map displayed over there, that the OXOX, SOX2, and KLF4 motif pairs, which are the known pioneers in the existing literature, indeed are cooperating. And interestingly, though, when you look at the relative distance between the motifs, when two motifs are further than about 150 to 200 base pairs apart, they stop cooperating at the level of accessibility, suggesting that this kind of cooperativity in order to remodel chromatin occurs at intra-nucleosomal distances. We've previously shown this with relations to transcription factor binding, but here I specifically show it at the level of accessibility. That both binding and accessibility cooperativity are distance dependent over the same distances indeed suggests a relationship between pioneering and the kind of downstream cooperativity that promotes transcription factor binding. So I show that all the motifs of pioneers can cooperate. So the next question would be whether low affinity motifs can cooperate to increase accessibility. As a showcase example, I show two motif pair of the OXOX and SOX2, two canonical pioneering motifs, and I break them up based off of their relative motif affinities and the relative distances to one another. Here you can see at the bottom, the lowest affinity motif pairs on the heat map are cooperating at levels that honestly rival that of high affinity motifs, and that regardless of the affinity of the motif pair, they are all limited to intranucleosomal distances. These results would suggest that pioneering cooperativity is widespread despite affinity and always distance dependent. One thing that really surprised us, however, is if I plot the observed accessibility reads given the differences in affinity and distance, we were surprised that the highest affinity motif pairs at the closest distances did not impart overall more accessibility. And the fact that our models were able to pretty accurately predict these relative accessibility levels indicated that this was indeed sequence encoded. So the lack of correlation was interesting to us, and we chose to interpret it that in vivo context is more complicated than the cooperativity of a single motif pair. Indeed, enhancers often have sometimes up to 12 different functional motifs that are bound. So we decided to look rather than in silico representations or a single motif pair and breaking things down, we also wanted to look at the in vivo context and how the motif behaves there. So I showed, just as an example, kind of my in silico simulations that I showed earlier as a reference. But instead now, I took every single mapped motif of the OXOX and SOX2 motifs, and I mutated them within their relative sequence context and took the log fold change effects of the wild type versus the mutated state. Here, we can immediately see, these are the exact same motif sequences as I showed above, but we can immediately see two very interesting things. For the SOX2 motif, in silico, it was very weak, but pretty canonical. But suddenly it becomes a very strong pioneering event amidst its in vivo sequences. Another thing that was interesting is that motif affinity for at least the SOX2 motif and to some extent the OXOX motif was a poor predictor of binding and accessibility strength at in vivo sequences. And this really surprised us. We thought affinity would have much more of an impact on whether transcription factor binding motif would be able to impart strong pioneering function. So I wanted to say, well, if it's motif organization that's actually making a motif a strong pioneer, then what kind of sequence features would actually be part of this organization? Because my previous slides have shown, again and again, that pioneering is inherently cooperative and distance dependent, I hypothesized that motif proximity to other strong pioneers might make itself a strong pioneer. Thus, I took every genomic region that we were interested in. I identified the strongest pioneering motif based off of the accessibility contribution scores from our accessibility model. And then for all the secondary motifs, I calculated the relative distances to see how they changed in terms of binding and accessibility effect, given how central they were to the strongest pioneer in the region. Here I show that unlike the scatter plot at the above where we can kind of see a breakaway from that three-way correlation, when we instead assess distance from the strongest pioneer, we can see actually a reformatting of the three-way correlation, kind of suggesting that like motif organization is an incredibly important part of whether transcription factors are able to impart a functional role on chromatin. And if I just quantify this differently and fit a linear model to it and take the residuals, the percent variance explained by the distance from the strongest pioneer actually outperforms that of PWM score directly, though affinity still does play a predictive role in determining whether it will be a strong pioneer. In order to experimentally validate this, because this shook us to our core, we were in an existential crisis. So we are now performing CRISPR experiments on a favorite region of interest that has 3 SOX2 motifs, 2 of which are high affinity with the exact same genomic sequence, and 1 of which is low affinity. If we look at our accessibility model, however, even though our experimental data shows that all 3 of these motifs are functionally bound using our ChIP-Nexus data, while the low affinity SOX2 motif is cooperatively pioneering, the high affinity distal SOX2 motif on the left is not thought to be a pioneer because of its suboptimal arrangement in this enhancer. And just to show as a control, our binding model, like our experimental evidence, does think that this is being bound by SOX2. So we want to go in, we want to make targeted mutations across these SOX2 high affinity motifs, just to validate that in vivo we are observing the same kinds of things that our models are interpreting, and that, you know, we're actually finding the right grammar. So taken together, this is kind of a showcase example of once amazing computer scientists come up with these neural networks, they're stable and they can be well trained. Here are the kind of sequence rules that we can extract from these neural networks. So in summary, low affinity motifs can be functionally mapped using deep learning. They play an important cooperative role in pioneering chromatin. Pioneering cooperativity is limited generally to intra-nucleosomal distances. And interestingly, in vivo motif organization is a stronger predictor of pioneering capacity than motif affinity, at least in the context of like the 2 to 3 pioneers that I'm considering in mouse embryonic stem cells. So I'd really like to thank everyone in our lab. This is highly interdisciplinary work. Without the experimentalists, I would be absolutely worthless. So thank you so much to them. As well as thank you to the Kondaji Lab for developing these interpretable deep learning methods, and the Stowers Core facilities to ensure that the experiments actually get done to validate what we're trying to learn using our deep learning models. Thank you.
We only have time for one question, just keep us on track.
All right, yeah, quick question. This is great work. In terms of pioneering activity on the protein side, it is also, I think, thought to be correlated with the concentration of the protein itself,
yeah.
right? So is there a good way to incorporate that information into these models? I mean,
yes, absolutely. So I actually didn't have time to show you the slide, but something we're also doing in the work that we're publishing as a preprint is I actually trained ChromeBPNet models over changing concentrations given the Patrick Kramer paper published in 2022 for OCT4. And what we found, it's just fascinating. So basically, what we found, this is a great question, I'm so glad you asked this, is that low affinity motifs really do rely on their surrounding sequence context in order to be functional. And that, for example, low concentrations of transcription factors, if low affinity motifs are by themselves and not clustered around other groups, they immediately drop off and become non-functional. But if they are in highly grouped regions with other pioneering motifs, they are able to stay functional at lower TF concentrations, we presume because the cooperativity helps bolster their impact. So it's really like concentration adds another degree of complexity and regulatory fine-tuning where you kind of balance affinity, optimal organization, and the relative concentration. So we do have work validating that. Yeah.
Good.